talk maybe very briefly about what problems, maybe not what problems, but what are the advantages and some problems of nanotoxicology, modern application, and so on. Again, just for you not to forget, tests today, consultation after lecture, and exams this Monday with 2 tests. There are some advanced, let's say, approaches to assess nanotoxicology, and these approaches united under the term omics. So omics means something in whole. So it's just some term plus omics. And the first one is transcriptomics. How the transcriptomics works and what it's assessing and so on. So as you probably all of you know, it's a central dogma of biology. The DNA upon a transcription process go to the RNA, and upon a translation process there is a synthesis of proteins. So genomics now, it's well studied, and all other things are also well studied, but since everything started with genomics, and in maybe recent decades, transcriptomics, which assessing RNA, is become more and more widespread. And how this works and why this is so important. Well, the general principle of transcriptomics is that you have some tissue samples. It doesn't matter which one. Here it's a fish. You have all of the RNA extracted, you perform some isolation of RNA, fragmentation, you form a library, you make a whole sequencing of this library and obtain some transcriptome assembly, and after this one you have a library with different expressions, so like it's called heat map. And why is this so important? Because, for example, if you're going to assess some cytotoxicity of something and you have, for example, some decrease in some proteins, for example, some protein level decreasing, you cannot understand for sure what is the real cause of this decrease. It's just a decrease because of oxidation of these proteins, because of adsorption of these proteins on surface of nanoparticles. So is there any other causes for this decrease? And in some cases, this decrease can be associated with damage done to RNA, so to translation process. And the answer to this question, why this happened, can be transcriptomics. So if you see that some of the genes are underexpressed or overexpressed, this will be the answer. And there's now a lot of papers, very recent papers, that working with transcriptomics approach. In some cases, there are even some cross-linkings between transcriptomics, genomics, and toxicity. And for example, in this paper, it's called Toxicogenomics approach. And in this particular paper, all of the study were conducted on nematodes C. elegans, which this is a very frequently used model organism because the whole genome of C. elegans already sequenced, and everything is known about this organism. So every gene is known, what is the purpose of these genes, and so on. And in this paper, also studied the impact of iron oxide nanoparticles, and they screened all of the genes and they measured the levels of these genes. And that's why they show how this can, how there are nanoparticles in form of citrate-coated superparamagnetic iron oxide nanoparticles, so BCA-covered spions, so how they can alter the cell viability of some of the cells from C. elegans. Well, in some cases, this is a very powerful approach, but of course, it's need a lot of equipment, a lot of work, and a lot of attention, and so on. And to be honest, if your lab or you are able to perform a whole sequencing of a whole C. elegans genome and form a transcriptome test for this worm, it's Well, not in all cases this will be a good paper, because this one published in Nanotoxicology and probably received maybe 100 or not 100, just 10 or something like this citations. Well, citations, of course, it's not a main measure of scientific impact of the paper, but still, not all of the transcriptomics approaches are so important or powerful and so on, and we'll discuss why this is so. The second approach, as we see from the Zin's dogma is proteomics. So what is a proteomics? It's analysis of well, a whole proteome of the particle of the cell. In case of proteome analysis, well, in some cases when you perform some fluorescence staining or metabolic assays and so on, you can analyze just a part of the proteins. So you can perform some immunostaining and so on. And this will result in analysis only just of one protein. But there's hundreds, thousands maybe of proteins inside each cell, and no one knows what happened to other proteins. And to perform proteomic analysis, you will usually need some mass spectroscopy. So what is mass spectroscopy? As you can see here on the scheme, mass spectroscopy usually works with some electron sputtering. So you have some sample, organic sample, inorganic sample, well, usually organic sample. This sample evaporated. After this, it's accelerated into magnetic field, going through some magnetic separators to a detector, and detector will detects based on molecular weights and charges of these molecules. And according to a specific database, some of the information can be absorbed in form of a mass spectrum. And this is mostly in case of some low, maybe not low molecular weight compounds. But if we're talking about some proteins, polysaccharides, peptides, and so on, in most of the cases we need another type of mass spectroscopy called MALDI-TOF. So MALDI, it's a matrix-assisted laser desorption ionization, and usually, as you can see here, it's look like a big cylinder, and MALDI-TOF devices looks like a big box, like 2 meters high, 1 meter width and length, and so on. So it's occupying usually half of the room. And the main difference between just a mass spectroscopy or how it's called, electrospray ionization mass spectroscopy and MALDI-TOF spectroscopy, the main difference in ionization power. So for proteins and polymers and so on, you need much lower power not to evaporate, not to destroy all of your sample. Well, in total you will have a spectrum, and according to the spectrum you can almost sequence all of the protein from your sample. Of course you need some sample preparation, you need some protein separation from your, for example, cell lysate and so on. What's in the result? As a result, you will obtain a whole protein, proteome, from a cell, some type of cells, and you can use it to study something. It depends on your research, of course. One of the applications for proteomics is studying of protein coronal structure, and as you can see, for example, here, it's a surface proteomics from nanoparticles, And using this proteomics approach, you can, for example, as this, for example, article states, prototyping of nanoparticles. So which nanoparticles absorb each protein and so on. But again, it's published in Nanoscalar Horizons. Sounds very good, but received maybe less than 10 citations, and that's because, of course, it's looking very nice. It's very complicated approach again. But to be honest, it's not that necessary in most of the cases to know the precise structure of protein corona, because, well, okay, 10% of protein corona, it's some kind of this kind of protein, and what's next? What's the purpose of this research? How it can be utilized? How it can be, I don't know, how you can engineer these nanoparticles to prevent this desorption, or how can be a useful application for this approach. So it's just a study to study. It's like, can be called because we can, and that's all. But what's next? So it's transcriptomics, proteomics, and, well, the next stage, obviously, it's metabolomics. So metabolome, it's everything that can be synthesized by a cell or an organism. So it's genomics, as I said, transcriptomics, proteomics, and metabolomics. And additionally to this one, there's other types of omics, called, for example, lipidomics. It's a studying of all of the lipids, and glycomics, it's a carbohydrate study. And what's so special about metabolomics? Well, in most of the cases, you still need mass spectroscopy, because most of the metabolites, it's rather proteins or some small molecules. Maybe vitamins, maybe some other hormones, and so on. Amino acids can be also. And what is the result of metabolomics approach? Again, this one quite a good article. Why? Because it's a studying of it's an application of metabolomics approach to studying of titanium dioxide nanoparticles and its toxic impact on rice. And rice, it's before we only discussed different cell models, animal models, and toxicological studies in studies connected with plants. They are also very widespread, because, for example, in Asia, it's a lot of people, most of the people eating rice, and this is the number one, if I remember correctly, number one plant culture in the world. So it's very important to study how the impact of the most produced nanoparticles, again, as I remember correctly, titanium dioxide nanoparticles is the most produced in the world now. So how the most produced can affect the most produced. And what is the result of the metabolomics study? The results may be presented in some of the plot charts and so on. And on this chart, you can plot a lot of different metabolites. Again, you cannot study everything, because it's just important to study everything, especially if it's only a plant, not even an animal or some insects or something like this. But still, you can plot some chart, and more important, after this one, you can plot some connection between metabolites and understand which ways are affected by nanoparticles. So what are the toxic pathways of this titanium dioxide when they apply to rice cultures? And when you are using just a simple approach, for example, some fluorescence staining, of course, you can take some leaves, you can take some other parts of rice and absorb some, I don't know, some oxidative stress in these leaves, in some other type of cells, but it would be impossible for you to understand why this happened, or maybe you will observe some decrease in production of some valuable amino acids, but you will not able to understand why this happens. And to understand, you need some kind of metabolomics approach to plot this graph and to observe what really happened. and that's why it's very, very powerful. But if we go even further, we can, and it's called multiomics approach. And multiomics approach, it's when you're using not just a metabolomics approach, but you also use some additional like transcriptomics approach, and usually it's like a proteomics plus metabolomics. And in most of the cases, it's again it's a mass spectroscopy. As you can see here, most of these techniques using mass spectroscopy, and in case of transcriptomics, it's a cDNA microarrays and so on. But it's a lot of most of this. It's again C. elegans and all of us. To be honest, I don't know what this strain is, but I think it's some kind of earthworm. But again, it's a whole organism. But all further, it's a human cells. Yeah, human cells. So what are the results? I mentioned that this is a very powerful approach and so on, very powerful and very expensive. To be noticed, but if you look on main affected pathways, in most of the cases the results is obvious, because, for example, oxidative stress, intracellular stress, energy mitochondrial function well connected with oxidation stress. Well, cell structure and so on, oxidative stress related pathway. Well, photosynthesis. Oh, okay, photosynthesis. It's something connected with maybe it's some unicellular algae or some cyanobacteria or something like this one. It doesn't matter. In most of the cases, the result is obvious: oxidative stress, but inflammation and so on. And well, sometimes it's not worth to use some multiomics or metabolomics approach if the result is obvious for you. So, well, maybe it's important if you have some advanced studies, if you are intended to use these particles in a large-scale production, which in some ways can affect our environment, or you are really going to use this in humans as drugs and so on. But in most of the cases, some of these results can be obtained very easily just using some fluorescent staining, some in vitro metabolic assays and so on. So yes, it's powerful, but you should not hope to obtain something outstanding using this approach. It just will give you more details and of course consume a lot of more money. We stopped at multiomics, and well, there's other type of interesting approach that can be used in nanotoxicology, despite it's connected with something molecular, and well, it's called molecular dynamics. So in most of the cases, molecular dynamics used to assess some of the proteins and connection between proteins and conformational changes of proteins, other molecules, and so on. But in some cases, you can use this to check how your proteins will interact with nanoparticles, not even the proteins, but some peptides, maybe some other molecules, and so on. And in some papers, molecular dynamics, which is already in silico methods, so there is no use of living organism at all. And to be honest, there's a lot of other types of in silico methods in nanotoxicology, but most of them are so far from real practice, and most important, some of them are very far from real life, because you can calculate using your computer almost everything, but not everything can be repeated in real life. Yes, it's a good option considering some ethical problems using animals and some other type of in vivo ex vivo models. But in some cases the results are not very realistic. And for example, here is a summary of some literature data, recent literature data available on the interaction of nanoparticles with different proteins or peptides. So it's in most of the cases it's a graphene or carbon nanotubes and so on. And why? Because they are well studied and there's some already existing models of carbon nanotubes interacting with proteins and how they can change the conformation, how they can interact and so on. Well, looking nice, maybe you can publish this in some high-ranked journals, but well, a lot of studies were dedicated to interaction with carbon nanotubes with amyloid beta peptides, which can lead to Alzheimer's diseases, and well, a lot of papers were published considering this one, and we will discuss it a bit later about how this can be really true. And to understand what is the perspective of using different technologies in vitro studying, this is a recent report published this year almost. And there's a lot of perspective can be put in, for example, omics technology, because yeah, there's some growth. Definitely some growth, not maybe very big. But if we are looking on current year and 7-year perspective, it may be from 2 to 3 times growth in omics technologies. And in other technologies also true, and as you can see, there's overall growth in vitro testing. And if we're looking, it's a lot of in vitro testings in pharmaceutical industry now. No, not only on not in pharmaceutical and cosmetics industry, because there's a lot of talks about using animals in cosmetics testing and so on. So now scientists trying to add some more in vitro testing, especially in cosmetics and so on. That's why it's a lot of things will happen probably in coming years in vitro testings, considering more advanced lab-on-a-chip technologies, maybe some omics technologies connected with lab-on-a-chip, or other 3D testing cultures, 3D spheroids, maybe some advanced in vitro models and so on. And that's all very good, but there's a further advance of further, let's say, application of nanotoxicology. We talked on the level of separate organisms. So transcriptomics, metabolomics, it's only one organism. But what can happen if this organism will come to nature, or you spill your nanoparticles in some, I don't know, lake or river or something like this one, and here comes environmental nanotoxicology. And when we are talking about environmental nanotoxicology, someone called it like a nanoecotoxicology and so on. But when we're talking about ecotoxicology, most of the guides and most of the protocols and methods in ecotoxicology are not very suitable for testing nanoparticles. For now, the basic models of ecotoxicology, usually it's like fishes, like a zebrafish, Danio, and some other water organism like Daphnia magna, or some earth organism, as I already mentioned, it's a C. elegans, and other very primitive but still in vivo systems. And these in vivo systems, well, they all of these base models, they are very well studied. Scientists know everything about this organism. But when we are talking about nanoparticles, usual toxicants, they're usually molecules. So molecules of some kinds, or maybe some metals in ion forms. Maybe it's even some kind of, I don't know, pesticides or some molecular compounds. But nanoparticles can go in other ways in real system, and it's connected with toxicogenomics. So scientists, especially toxicologists, ecologists, they don't know how these nanoparticles can behave in real life. So for example, in lake, what will happen with these nanoparticles? Because nanoparticles can undergo some aging process. They can decompose in other, not in other nanoparticles, not in other elements, but they can oxidize, or maybe they can dissolute. There can be some light-dependent reactions, and if you take some, for example, probe of water, it's already a question where to store it to absorb some reactions. So nanoparticles can absorb nutrients on its surface or inside these nanoparticles, and this also can affect some viability of living organism. And there's also a question, what are the concentration of nanoparticles in this water, for example? What methods you need to use to assess this? What are the routes of exposure of this living organism to these nanoparticles? And there's a lot of questions. And because ecologists are dealing with different molecular toxicants, in most of the cases, they completely don't know what to do with nanoparticles, because just imagine, sometimes, especially those of you who already worked with nanoparticles on scanning electron microscope, sometimes it's hard to find your nanoparticles, even if you know that there should be some particles, because you dilute them to a very big extent or something like this one. But imagine to find some nanoparticles in a lake, or in a river, or in a whole fish. So you need to perform a lot of investigation to find something. You need to dissect this poor fish in slices, a whole fish completely in slices, or you need to test every organ to find where these nanoparticles accumulate and how they affect the viability of this fish. And what's so important, what will happen to a human if it will eat this fish? Because of this one, it's a lot of critical issues with test guidelines according to connected with ecotoxicology. Because of course most of the particles will agglomerate and aggregate when they emerged in our environment. There can be some dissolution, agglomeration, a lot of question with effective doses. So particle concentration, what can happen to coatings of these nanoparticles? Will they be coated with some additional environmental molecules, or opposite, will they lose this coating, and so on? And because of enormous amount of questions on this stage, there's enormous amount of physical and chemicals criteria, and a lot of testing problems connected with testing of these nanoparticles. And a lot of scientists now trying to develop some criteria to test these nanoparticles in environment, because as you should understand, it's a lot of physical and chemical properties of engineering nanoparticles. And in laboratory conditions, even we as a scientist not always measuring all of these things, because maybe none of you ever measured the density of nanoparticles, or maybe some conductivity of nanoparticles, and in some cases it's very important. It's also important to measure all of this toxicity criteria of these nanoparticles, toxicity characteristics, to simulate, for example, freshwater condition or saltwater condition. And well, looking on all of this, it really looks very complicated, and because of this, it's very hard to formulate some let's say guidance, modern guidance to test nanoparticles toxicity in environment. And because of this complexity, now there's in recent years it's a lot of, maybe not a lot, but still some of the articles published which are intended to observe some toxic outcomes from naturally occurring nanoparticles. What are naturally occurring nanoparticles? It's nanoparticles which are formed not by only humans, but by nature itself. because it can be a wind erosion when wind lifts some particles from soil, from mines, coal mines, especially copper mines, and so on. And particularly this investigation, published in previous year, caused a lot of publication in papers in different media, because there was a very, let's say, not bad, but not very good results. It's published in Environmental Research Journal, which devoted to different environmental researches, obviously. And it's analysis from Mexico region, and there's a lot of different mines in this region, and there is a heart tissue of patients, and there was a lot of magnetic and magnetite nanoparticles found in heart tissues and brain tissues and so on. And what's most important, this is the tissues of a very young humans. So as you can see, average age of these humans are like 20s, and in some cases it's 40s. So even at this stage of our life, so a very, very young human, it's just 3 years old. And we already have nanoparticles spread around our organism. And the question raised in this paper and in other papers, because we are exposed to these nanoparticles from different industrial processes, maybe in some of the cases, for example, in case of Alzheimer's diseases, because the probability of Alzheimer's diseases increases in recent years, maybe some of our diseases are strictly connected with nanoparticles. Maybe it's not just aging of population or mutations or something like this one. And it's a good question after all, because there's a lot of nanoparticles now around us, and scientists still believe that these particles in general not very toxic. Maybe we already in I don't know, in past hundred of years already exposed to a lot of nanoparticles, and maybe these nanoparticles is a real cause of our diseases, and we need to do something with this. Maybe some air, more strictly air pollution guidance and so on. And if you look to mass media, you will see a lot of news published in every year in recent years because of nanoparticles from coal dust, even nanoparticles from 3D printers. So when 3D printers print something, there was found nanoparticles of plastic in the air. So it's not very healthy to stand even close to not bioprinters but 3D printers and inhale this air, because you can accumulate nanoplastic in your lungs. But to be honest, life is a very dangerous process, as you know, and if you will read all of this news, it will be not very healthy for our psychic condition to know everything about our environment. But still, you need to understand that there's a lot of nanoparticles around us, which can be originated from mines, even from some industrial processes, and even from our very close environment. And because of this one, for example, in Europe, in America, in USA, it's a lot of different frameworks, a lot of environmental protocols were published in past 15 maybe years, devoted to different evaluation and assessment of risks connected with nanoparticles. For example, Nano-Risk Framework published in European Union in 2007, and there's a lot of other published by different countries: Australia, Switzerland, USA, again Switzerland. A lot of things published in Switzerland. and Denmark, Netherlands, and so on. As far as I know, there is no such document published in Russia, because, well, we are only at the beginning of the assessment of this environmental pollution. But why? Because we are not we're producing not so many particles in large scale. And in Europe, if you produce more than, if I remember correctly, more than 1 ton of nanoparticle or something having nanoparticles in composition, you need to follow this guidance according to a country guidance. So maybe in coming years we will have some additional risk assessment documents, or maybe there would be some additional documents as was with genetically modified organisms, because, well, in some countries they are now prohibited. I hope this will not happen to nanoparticles, because it's a lot of problems for scientists. But it's not directly connected with nanotoxicology after all. That's why it was the last slide, and thank you for your attention. If you have any questions connected with nanotoxicology, because it's end of our course, you can ask me this question in chat. And by the way, answer to Alexei about XL. Yes, XL arrested in second metaphase, second stage of metaphase. But even if this XL is arrested, there is no way for this XL to perform, to complete this mitosis prior to fertilization. So it should be a sperm cell to continue this division, to continue this mitosis. So XL alone cannot undergo mitosis. That's why there was an option that this cell cannot. And after this lecture we will have a consultation. I hope Nastya asked me to present her topic during consultation, so we will hear for Nastya's topic. And after this one, you are free to ask me any questions connected with final tests, exam, or marks, or so on. So in maybe, as always, in 10 minutes, I'm waiting for you in Zoom room, following this link. And of course, you can ask me some additional questions there. So Alexei, you can find me after your test. Maybe I will still in Zoom room, and you can find me there, because we will have time until half before 7 p.m. So I can wait you there in Zoom room. If there's no questions, again, thank you for attention, and okay, I'm waiting. I'm waiting for should it be some other information? Okay. That's all. Waiting for you in Zoom room.